Antibiotics are overprescribed for a number of different reasons. Physicians in a busy clinic are pressed for time. They have a number of patients on their list of folks that they have to see that day. Patients may ultimately be coming in because they seek a solution to their problem. And antibiotics may be that quick fix. Write the prescription, patient satisfied, they're getting a solution, doc satisfied that the patient's happy, patient then goes on and the next patient comes in. So there may be an artificial prescribing habit based on the efficiency of what it gains to be able to give a solution to a problem that may not necessarily be the appropriate solution to that problem. The other reason, for example, in hospitals, it may be that antibiotics are overprescribed because doctors want, or nurse practitioners, whoever's prescribing them, want to give the best medicine, the best care to a patient, and they don't want to miss something that's treatable. And bacterial infections are generally currently treatable. And viral infections tend not to have a specific antiviral necessarily, although that is changing. And so even if someone's presenting with an infection where it may be viral or bacterial, and if they're very sick, for example, certainly doctors are going to want to treat what's treatable with antibacterials. But I think that also, unfortunately, even for those that aren't very sick, may also tend to let doctors or encourage doctors to overprescribe because they want to do something that they don't really want to regret not doing something that they could have done to help that patient. Yeah, so in terms of from a patient perspective, I think a lot of patients do expect a solution to their problem, and I think a lot of patients are used to getting a prescription as that solution. And so it's challenging because it may be that if physicians are trying not to give antibiotics as frequently as they perhaps previously did, it may be that their patients are expecting it, and if they're not getting that as a solution, it may encourage further discussion, which is indeed prolongs that visit, which is fine, but that may ultimately encourage the doctor to go back and write that prescription. But really that's an opportunity for doctors and patients to have a discussion, an education center, a discussion with regard to the fact that antibiotics will not help a non-bacterial infection. And hopefully with time, patients will be educated in that regard. But certainly I think right now there's a component, certainly in the past, where it was a patient request or patient-driven a desire to help those patients in terms of getting what they want, which is the antibiotic prescription to whatever the problem might be. Having said all that, there have been a lot of campaigns more recently, and I think there is more and more education that's actually now working in terms of educating the general public about the fact that not all infections, in fact, are bacterial and need antibiotics, but there's still a lot more that needs to be done. So if antibiotics become less effective, and it's not really if, they are already less effective, if it continues and we have less and less of an opportunity to have available antimicrobials, which is definitely in the near future, the consequences are significant. There'll be significant morbidity and mortality. We already have examples, not so much in Canada, but certainly in the United States, where there are patients who are presenting with what would be previously treatable bacterial infections that now essentially have no antibacterials to which the organism is susceptible. And as a result, then you're forced to either use archived antibiotics that may not be as effective and that may have toxicities, or you have no options left at all to offer those patients. As a consequence, they're left with their own immunity as their only defense against their infection, and some, certainly as we know in days of pre-antimicrobials, they'll survive that infection, and others will unfortunately ultimately be significantly ill and likely will die from their infections. And so I think it's amazing if you look back at folks who've gone into infectious diseases. Most of us went into infectious diseases because there is a treatment to most infectious diseases, and there is it's a wonderful thing to be able to help a patient and see them go from being very sick to being very healthy. And it's really a turn to the whole pre-antibiotic era, in that currently in terms of where we're seeing these multi-drug resistant organisms come through, and certainly if it continues at this pace, we will be essentially back at that pre-antibiotic era. So in general, right now, what previously was thought to be easily treatable and not a big issue in infectious diseases, it'll become, again, that this is going to be significantly associated with significant morbidity and mortality. Currently, the most resistant organisms that we're faced with are multi-drug resistant gram-negatives, whereby essentially the very resistant organisms are essentially resistant to all first-line drugs, and some now are being resistant even to our second and third-line drugs, to the point where there are some organisms that are not susceptible to any known antimicrobial. Those would be right now our biggest threat in terms of ultimately causing significant disease. Now, these drugs, these organisms, fortunately, have been primarily in folks who are colonized with them, not causing significant disease in the majority, but some certainly have gotten urinary tract infections, and some certainly have been sick with bacterial infections in the blood, or with what we call sepsis and the like, and have had significant morbidity associated with those infections. So in terms of right now, the biggest threat or the biggest problem in terms of antimicrobial resistance would certainly be these multi-drug, or what some would call extremely drug-resistant gram-negative organisms. The general public right now may not necessarily appreciate the full impact of what may ultimately come of antimicrobial resistance. I think partly because there are still options available for these now more resistant organisms than what had been previously available in the past. And so because the average person has not yet seen face-to-face the problem of having an organism for which there are no antimicrobials left, I think right now most folks think, well, yeah, there's resistance, but there's always another drug available. And the issue is that that's true for some of our resistant organisms like MRSA and VRE, where there have been newer drugs provided to be able to treat those infections. But the pipeline in terms of newer drugs being available is very, as you probably all heard, is very dry, and there's not that much in the forthcoming future. And so while there may be one or two options currently to address those resistant organisms, once we have development of resistance to those newer drugs, we may not have any drugs left. And yet the average person right now probably doesn't necessarily know that subtlety, and they may right now say, well, yes, I've got MRSA, but it's treatable. And so because it's still treatable, I don't think it's hit many persons that this is in fact a problem that may not be treatable in the next decade or so. Certainly those families that have been hit hard with these extremely drug-resistant organisms, they've certainly understand the concern because they've had that personal experience. But again, the prevalence of those extremely drug-resistant organisms is just now starting to pick up. And until I think more and more persons have had that first-hand experience and have seen a loved one who's very sick who ultimately dies from one of these infections, I don't think it'll necessarily hit home just yet. And one of the key things to be able to ultimately try to control these antimicrobial resistant organisms is to be able to, well, there's lots of different key things, but is to be able to, one of them is to detect them. And if you can't detect these antimicrobial resistant organisms, you won't be able to ultimately be able to manage them from a clinical point of view and prescribing the appropriate drug, and you won't be able to institute perhaps the appropriate precautions that are needed to prevent the spread of that organism to other patients if someone, for example, is an inpatient. One of the key important aspects to be able to control antimicrobial resistant organisms is to be able to diagnose them and detect them in the lab. To do that, there are lots of different technologies available, and certainly one of the newer technologies is rapid diagnostics. One example would be nucleic acid amplification testing like PCR. Other examples would be some of the newer chromogenic tests that allow for fast detection of enzymes that are present. For example, carbapenemase producing Enterobacteriaceae have a broad-spectrum beta-lactamase that breaks down carbapenems and beta-lactams. And there are newer tests now whereby you can add your organism to a broth that contains a chromogenic antibiotic, and if that organism is producing the beta-lactamase and breaks down that chromogenic antibiotic, it changes the color of the broth. And so within a few minutes, you can detect whether or not that organism might contain that carbapenemase, or, for example, a similar beta-lactamase like ESBLs. There are also newer tests looking at detection of underlying proteins like the altered penicillin binding protein 2A with MRSA through something called latex agglutination, whereby within, again, minutes you'll know whether or not that mechanism is present in the organism. So really all of those tests, whether it be nucleic acid amplification testing, whether it be a protein-based assay looking for something through latex agglutination, or whether it be through these novel chromogenic antibiotics that will allow for detection of breakdown, they all allow for detection of these resistant mechanisms within a very short order of essentially minutes. And that certainly does help in terms of expediting getting a preliminary report out to physicians and treating teams that will let them know about the resistance, and will also allow for our infection control colleagues to institute the appropriate precautions to prevent the spread of that resistance. The challenge of rapid diagnostics on specimens as opposed to on isolates is that specimens may have more than one organism. And we know that our bodies are filled with organisms, our normal microbial flora, which we need and don't want to get rid of. And it does challenge diagnostic laboratories in terms of developing rapid diagnostics that will pick up resistance mechanisms, in that if you were to pick up, for example, the underlying gene for methicillin resistance in Staph aureus called MEK-A, if you were to pick a swab, let's say, that may be mixed with skin flora called coagulase negative staph, and we also have MRSA, which is the Staph aureus organism, we know that our coagulase negative staph may carry that MEK-A gene. So if you find the MEK-A, the question is, well, is it in the coagulase staph, which we actually are not as concerned about, or is it in the more virulent organisms of Staph aureus? We don't know necessarily. And so actually there are a lot of different companies that have put out rapid diagnostics looking for underlying genes, and the challenge that they're facing is if we do it right off of specimen, not off of an organism, it may not necessarily be easy to translate where that gene came from. Having said that, I think there still is a role for that. Certainly in terms of rapid screening for our carbapenemase genes, whereby we don't necessarily need to know which organism they're in, but if it's present at all, it's a concern. That's something that I think really will be something that would be very helpful and can be done certainly in some of the novel diagnostic methods with random access, real-time PCR, and basically within less than an hour, within 45 minutes or so. The challenge of some of these other mechanisms or underlying mechanisms that may be shared upon more virulent worrisome bugs and less virulent, not as worrisome bugs, is something that I think we'd have to face in that context. But yes, I think there certainly is a role for these tests. In terms of when will they be out there, well, there's a lot of companies right now that are producing some of these very easy to use random access multiplex assays looking for key resistance determinants. None are yet out in the market fully. They're just in development, and certainly all have some limitations in that they may not necessarily be as comprehensive and picking up all resistant determinants, and they may or may not ultimately be something that's amenable to being testing specimens directly. It may be something that folks would rather actually do on isolates specifically. So the challenge is where to actually apply them. Is it at the bedside, at the diagnostic level, or is it at the lab side where you have an organism and you want rapid detection of resistance at the bug level? So I think there's still some nuances that have to be worked out once we see those assays. Realistically, probably we're going to see them in the next 5 years out and available and being used. The question is how they be used, not yet quite clear. Okay, so in the clinic, if you were to, the ideal situation would be you have a patient in front of you, and let's say they're presenting with pneumonia, don't need to be hospitalized, they're sick but not as sick enough to need to be hospitalized, and you'd like to know whether or not you can identify the pathogen and also identify whether or not it may be a resistant pathogen. For example, Streptococcus pneumonia, most common cause of community-acquired pneumonia. Well, is it pen resistant? If not, you can use penicillin. If it is pen resistant, is ceftriaxone susceptible, and the whole issues, or azithromycin and erythromycin have all those mechanisms. The challenge is it's not something that is as easy as just one mechanism. There's multiple different organisms that can cause pneumonia, and there's multiple different mechanisms of resistance that may develop within one class, let alone multiple classes. So it's not as easy to think about having one black box diagnostic method that will be able to pick up the common causes and also have the resistance determinants all in one easy kind of component, especially when you're dealing with specimens like a sputum sample, which is going to go through the oral flora and pick up all the normal bacteria as well, which may actually carry some of those resistant determinants. So that's the real challenge, is that ultimate ideal would be in the clinic, you've got your bugs and you've got your resistance determinants both identified in a very quick one-stop shopping assay. And yet it's not a pure sample there. You've got a person giving you an expectorated sputum through an oral pharynx that's got normal healthy bacterial flora that are not causing the problem. So if you were to pick up, for example, Streptococcus pneumoniae, and you pick up that it has the gene, which basically causes it to be resistant to macrolides, you couldn't use azithromycin to treat that patient. The question is that gene in the strep pneumo, or is that gene in the virulence group strep that's in their mouth that's not causing them to be sick? And that's the challenge, is how to link the gene that you found to the bug that you found without having to grow it in the lab and actually multiply the organism and find it. And I haven't seen any company yet that's been able to do that link. I'm sure there will be the ability to do so based on very good, accurate counting of copies of DNA to be able to actually link genes with actual genes from that particular organism. But I haven't seen it yet. So I don't know, I think that's kind of an ultimate goal. I'm not sure when we'll get there. Right now, certainly though, having rapid diagnostics in terms of detecting organisms without the genetic component to detect resistance is something viable. Bringing it to the lab is still the norm. We're not really doing any bedside diagnostics at this point for organisms or for resistance. Realistically, what the next kind of step is for labs would be to get specimens to the lab, identify the organism rapidly. So we still culture, but we have novel methods now to detect identification of organisms. The biggest one now is called multi-TOF mass spectrometry. And that really now allows us to identify bugs the same day that they're growing, even if they're only growing in small amounts. So we're getting much more rapid diagnostics in terms of identifying the actual organism that's causing the disease. What's lagging though is how to do rapid understanding of resistance. And so some of the methods I mentioned before with the chromogenic beta-lactam drugs that will be broken down by carbapentamases are helping us to get out preliminary reports out faster for presumptive ESBL or carbapenemase producing organisms. The other methods I mentioned in terms of looking for protein-based mechanisms are also helping us to have that same day as the organism goes out, we're given a presumptive presence or absence of those mechanisms. But for the majority of mechanisms of resistance right now, there are not assays, and that it is waiting for the bug to actually grow with the drug overnight for the next day to get your results. And that's a huge area that would be lovely to be able to expedite and getting that fixed so it is same-day diagnostics within the lab. Right, so I think the clinic issue is how do you link then an underlying gene and link it to the actual organism that you've also detected genetically. And the challenge is in the clinic, most folks aren't sick, are not that sick, and you're going to be getting samples that they can produce easily, which are going to normally be going through orifices that have other normal bacteria. So you're always dealing with a contaminated sample when you're testing. And the issue is we need better diagnostics that are more accurate at picking up actual quantity of organism, then that they can then ultimately link, well, we have a gene that's in the same exact quantity of that organism. It makes sense that they're linked because the virulence group strep assay is not picking up as much virulence group strep, which means it doesn't make sense that that organism, that gene is actually from that particular organism. So that's the nuances that have to be done before we would see the jump to clinic of getting both a bug and a resistant mechanism. Having said all that, there are lots of companies out there trying to do that right now. I haven't seen a lot that have linked them. They have great multiplex assays to detect bug and detect resistance determinants. The next step is, well, how do you link those together? And that's really the challenge right now that a lot of companies are facing to really make it the next big diagnostic tool that will be used in clinics. One of the challenges in clinic is knowing when you have a bacterial infection or a non-bacterial infection. And that is a subtle difference. You can't make that diagnosis necessarily clearly based on symptoms only. And that is one of the reasons why physicians may overprescribe antibiotics, because they don't want to miss the treatable cause of what the person's presenting with. And so ideally, those rapid diagnostics would be introduced in the clinic in that capacity, not so much to detect resistance as much as simply detecting the culprit organism. And if indeed it's clearly an adenovirus or influenza virus or rhinovirus or coronavirus, and the patient symptoms are compatible with that virus, then the antibacterials aren't going to be prescribed. And instead, perhaps would be more appropriate to prescribe antivirals for the influenza virus as an example. And so having those types of rapid diagnostics available immediately at the bedside or in the clinic itself would be something that is hugely advantageous. And there are right now rapid nucleic acid amplification tests for influenza and for some of the respiratory viral diseases that are incredibly rapid. So right now, for example, in our laboratory, we're using one that's random access, real-time PCR, the instrument that has the capability of having assays for influenza and RSV. And essentially results are within an hour or less, 45 minutes to an hour, of getting the actual sample to the lab. Similarly, there's another assay that's out there which has a very comparable, almost random access capability within a 45 minute to an hour turnaround time. Right now, those are in labs. And if you can get the specimen to the lab quickly and your patient doesn't mind waiting, you can still have that same day discussion. Alternatively, if you have that capacity in your lab that supports your clinic, you could say to the patient, this most likely is viral, and I've taken a sample, and I'll let you know. And even if the test is negative, I still think it's viral. Let me know how you're doing in a few days. And if you're still having problems, come back and I'll see you. Or they may write a prescription saying don't fill it unless you're still X, Y, and Z sick, and then give instructions, for example, in a few days' time. One of the challenges of these assays is the sensitivity or negative predictive value. That if you have a negative test, well, does it mean that you don't have a virus that's causing it? There are so many viruses that can cause the common cold and pharyngitis and the like. And so you don't want necessarily doctors to become dependent on finding a virus to say it's viral and prescribing antibiotics for any time it's negative, because it may just be that the assay is not looking for all viruses, and maybe the sample itself was not a good sample and didn't pick up a lot of cells with the virus, or maybe the patient's presenting so late that the amount of virus is so low that the assay won't pick it up. And so you introduce these into the clinic, you want to make sure there's a lot of education, that certainly it's helpful to look for flu and to treat if it is flu, but a negative won't necessarily rule out that it's not another viral potential organism that's causing the actual syndrome itself. So that it has to be nuanced, and education has to be certainly a part of the introduction to make sure that docs don't just prescribe for every negative viral assay antibiotics for presumed then bacterial cause, because we certainly know that that would not be appropriate either. Surveillance is key to understanding antimicrobial resistance. If you don't have surveillance, you won't know when you have a new resistant organism in your population. And at the nation level, at the international level, and certainly at the local level, at all capacities, it's important to be able to know. The issue of, well, what are we doing here in Canada? We actually just had a review at our last Medical Microbiology Infectious Disease Conference last year, looking, it was actually a research project of an individual who looked at all the surveillance systems in Canada. And essentially was trying to answer the question is, are they sufficient, or do we need to have a better overview of what's going on? And right now there are a number of different systems. The main ones right now that we have in Canada, one is called CanWard, and it uses sentinel laboratories throughout Canada, involving almost all regions, that provide basically random sampling of their isolates every year to this one central laboratory. There's also something called CNISP, which is the Canadian Nosocomial Infection Surveillance Program. And they're really the major nationwide surveillance programs that we have. There are others. There's a Toronto-based program, Toronto Invasive Bacterial Disease Network, it's called, or TIBDN. There are some industry-based programs that are international that have a Canadian component. One is called SMART, another one's called TEST. Each has their own specific focus, of course, and each has their own pros and cons. And ultimately, the question is, well, could there be an improved system? I think the resounding answer would be yes. One of the issues that I find troubling or hard to deal with as an infectious disease physician and a microbiologist is knowing what's going on in the community in terms of especially novel resistant organisms. And one of the aspects that is challenging is that right now there's no mandate to report what you have found in your lab. There's no reporting system. We do have mandatory reporting systems for key infectious diseases that have a potential public health impact in terms of their communicability in outbreak situations, but we don't yet have a reporting system for antimicrobial resistant organisms, so that there's systematic surveillance going on in real time of key resistant organisms. And part of that is that there are so many now resistant organisms, I don't think we'd have the capacity to be able to track them all in real time. But certainly for the novel ones, like the carbapenemase producing Enterobacteriaceae right now that we're all seeing, and for other extremely drug-resistant organisms as they are introduced into different locations, it would be obviously to everyone's best interest to know that they've been introduced into a population, especially one whereby you may start seeing patients from that population. And the communication to the public at large and to physicians and epidemiologists at large, I think, could definitely be improved by having mandatory reporting, and also by having, ideally, some systematic way of having a reference laboratory have access to these isolates so that they can understand how related they are to one another and what the impact might be ultimately on treatment and the like based on their underlying genetic resistance determinants. So I think right now we have a lot of these programs in place, which is fantastic, but they're not comprehensive and they're not necessarily in real time. They often will have the reports out the year following, and they don't necessarily report out based on locality to let you know right now that there's a specific problem going on in this part of Canada. Right now, what people depend on is really word of mouth. The epidemiologists or infection control persons in hospitals all tend to have networks and discuss with one another. But from a systematic way of reporting, especially to the public and to physicians who aren't part of that network, it's not yet there. There's been some improvements. In Ontario, they have mandatory reporting now of rates of MRSA and VRE and C. difficile in hospitals, so that at least there's some familiarity with which hospitals may be having concerns or outbreaks. But it's not, again, looking at the issue of the novel resistance determinants and the sharing of resistance data from one hospital to the others. Some people have solutions. Ourselves, we right now post all of our antibiograms online to share that with the community. We're looking at improving that in terms of making these antibiograms searchable and comparable with data that others can actually upload themselves. And we're doing that for a number of reasons. One is to allow for our large cohort of client hospitals to have access to the data, no matter where they are, but also so that we help facilitate those community hospitals that may not have the capacity to do their own antibiograms to at least have some familiarity of resistance that's going on in the Greater Toronto Area hospitals. One of the challenges is each clinic, each hospital, essentially has the option to choose whatever electronic medical record they so choose, and so there's not a commonality necessarily between them. So the software may differ in one clinic versus another versus one hospital and the like, so the communications can be challenging in terms of interfacing that. There is a project right now in Ontario called OLIS, which is the Ontario Laboratory Information System, and they are currently working with different hospitals, and it's at the hospital level that they're working, not at the clinic level right now, as far as I understand. And they are interfacing with each hospital system so that that data from that hospital can be uploaded to their common database. And then this common database is available to physicians and treating teams to access. Now, their rationale is not so much for surveillance. Right now, it's so that you can, the physicians and treating teams can access information of a patient who was admitted at a different hospital than yours, so that you don't have to ask for their medical records to copy their chart, etc., and you can readily access both lab data and other data from that other hospital. It's not where it needs to be at yet to be something that would ultimately be useful from a surveillance point of view. They're still working with hospitals and getting those hospitals updated to be actually interfaceable. As to whether or not that may be a potential, I don't know the capacity of that software to know whether it can then go ahead and data mine to find the resistance easily and produce nice antibiograms. If it were, that would be fantastic in terms of doing it at a community level. Mind you, it's specific only to Ontario. The reality is, ideally, you'd see that the nationwide basically system, so that you're getting data in real time from multiple different regions, as opposed to just within one region. But nonetheless, it's a reasonable start. But I just don't know the capacity. It's taken a number of years for it to get where it's at now, which is simply getting the actual data from each hospital to its software. The data mining capacity needs to be explored further. It's an excellent suggestion to have data mining essentially of electronic medical records. Certainly right now when we do our antibiogram development, we go to our personal laboratory information system and we extract the data from there. There is a lot of cleanup that you have to do to that data to massage it out and really get accurate resistance rate estimates. And so it's challenging to try to make that automated. We're trying to right now do as much as we can with a computer expert. Whether we get to where we want to be, we'll see hopefully in the next year, which is our expected deadline date for our project. Certainly the more automated things are, the more likely they're going to be available real time and available for use. And so I think it's a perfect goal to have. Haven't seen a system yet that's doing it. Is can we create a system whereby it's actually part of the expected mandate of every hospital to provide an antibiogram in a certain format that can then be ultimately uploaded to a common server and then shared. Certainly they've done that with regard to requesting all Ontario hospitals provide their MRSA, VRE, and C. difficile rates. The next would be, well, can they just provide their their antibiograms in general in a comparable fashion that can be uploaded and shareable and the like, so that geographically we can see where there are potential resistant hotspots. Yeah, antibiotic stewardship, it refers to essentially how to optimize the management of antimicrobials so that you maximize their benefit and you minimize the potential adverse effects. And those adverse effects could be, one, resistance, two, side effects of the actual drug that people may have, for example, allergies, three, complications like Clostridium difficile infection, which may occur by the use of antimicrobials, and four, ultimately, is cost. And so ultimately the idea behind those programs is, well, what can we do to maximize the benefit and the gain while minimizing all those potential negative adverse effects? So programs generally look at different tools they can use, and they don't use them all systematically, but they have them all available to them to use. One of which would be, well, let's start with looking at guidelines. If guidelines exist on how to manage these infections, let's make sure we at least are aware of those guidelines and follow them. Now, guidelines, generally speaking, say, okay, well, if the patient presents with X, Y, and Z, and you diagnose that they have a pneumonia, here's what we recommend to use in terms of antimicrobials, and they give options. So the next question is, well, which option should we choose? Well, you should look at your local antibiogram data. So most stewardship programs include now a request for antibiograms, which is going back to our discussion of surveillance, in that surveillance ultimately and knowing what's going on in your community is key to be able to advise the best empiric choice of drug. And so most folks now are providing at least a bare bones antibiogram to facilitate decisions about how best to provide guidance to clinical teams of how to treat certain syndromes. So it's really a combination of what guidelines are available, how to make those guidelines specific to your needs by looking at your local antibiogram data, and then ultimately, how do you implement this? You need to have education. And so these programs then are educating physicians and front-end clinical teams on understanding those guidelines, understanding ultimately how to look at your local antibiogram data yourself, and or how to actually look at the polished, improved guidelines that take the antibiogram into consideration. And ultimately what to do if the patient presents to your clinic and or to your emerge department and or to your ward, etc. Other options, if folks have already done that, or folks can't necessarily go through all of that challenge, those particular tools, would be to do specific audits and to look to see what prescribing habits occur in your facility. So they call these often audit and feedback strategies, whereby you either have a physician and or a pharmacist or both work on a specific key ward. Usually they start off in a ward where there's a lot of antimicrobial use, like ICUs, and they literally will go and audit every patient that had had an antibiotic prescribed. And they'll see whether or not that makes sense in their minds based on all their review of the data on that particular patient's chart. And they'll discuss it with the ICU team and essentially educate them on when they believe that the antimicrobials are perhaps prescribed in a fashion that is not needed. The antimicrobials themselves may not be needed at all, or they may be prescribing a very broad spectrum antibiotic, and a much more narrow one may be appropriate and all that you need. So they'll go ahead and make those advice, suggestions, and typically they're usually well welcomed by the team, and by doing so often they'll see a dramatic reduction in antimicrobial use that was inappropriate, and or a narrowing of that antimicrobial use itself. So that's audit and feedback. So we've got the guidance, the antibiograms, audit and feedback, and then there's other strategies, and again, it depends on the resources as to how many of these are used. But others would be to have automatic conversions from parenteral to oral antibiotics to try to reduce cost in terms of when folks can go into oral. And others will have conversions from broad spectrum to more narrow spectrum automatically at the pharmacist review level, again, to make more appropriate, more narrow spectrum antimicrobials to reduce the potential side effects. And finally, some may actually have restrictions of antimicrobials that cannot, in fact, even be prescribed without getting an approval ahead of time. And so all those strategies are used as part of the antibiotic stewardship programs that have now been implemented in all hospitals, or if not already, that are now being implemented, and that they have been mandated as being something that's required within Canadian hospitals. Part of that is the surveillance within your local facility, and that's not, again, a mandated component to stewardship programs, but the reality is it would be very challenging to have a stewardship program without knowing what's going on in your local community. So in that capacity, at least antibiograms, if they haven't been something that's been produced from hospitals in the past, are certainly going to be now produced. So going back to the discussion of surveillance and whether or not we can now tidy in those antibiograms to create a broader Canadian-wide antibiogram, certainly that's something that would be appealing to do. And again, some of those surveillance projects, just to go back to to CanWard and that's what their intent is to look at the Canadian antibiogram by getting data from labs they've actually went a step further of actually retesting the organisms to verify the resistance, to make sure that they are valid data. And it is an excellent program. The challenge is it's not in real time, and that's where it would be fantastic to have that real-time data. To address that, the big problem, antimicrobial resistance, is really not a one easy solution to go. There's multiple different angles you need to look at, and starting at the beginning is the indiscriminate use of antimicrobials, and from a physician and a human population point of view, it's the overprescribing of antimicrobials and the unnecessary use of very broad spectrum antimicrobials. And really the antibiotic stewardship programs that are entailed are being introduced at this point in hospitals are trying to address that issue. It's not yet something that's being introduced in the clinic level, and there it's education campaigns that are really trying to address that. That's really trying to avert the potential selection of resistance by reducing those issues of antimicrobials being present and allowing for that selection to occur. At the same time, parallel to the human population and the antimicrobials is the whole animal population, and that's really where policies have to be implemented in terms of the issue of restricting of the use of antimicrobials, whereby they're actually not used primarily to reduce infection, they're being used to allow for increased weight gain and growth of the animals themselves. And the consequences there, of course, are resistance on the food that we ultimately eat and then get introduced in that capacity, and or through potential remnants of the organism's fecal matter that may get into our water system and the like. So certainly it's a two-pronged approach of restricting the antimicrobials to actually reduce that selective pressure of actually getting resistance. And a bonus as well is that will reduce the adverse effects of antimicrobials in terms of C. difficile, and as well as adverse effects like allergies, and will also potentially reduce the cost associated with indiscriminate use. But once you've got resistance, the other big issue is how do you contain it and not allow for the spread? And within hospitals, that's where infection control and prevention programs are key. And that you need to have, well, one is the diagnostics to pick them up, and then two, the response to be able to then isolate those patients from other patients so you don't have the spread of this resistant organism from one patient to the next. And so that containment strategy of using contact precautions in isolation rooms and environmental cleaning is really a key component to be able to prevent the transmission of these resistant bugs from one patient to the next. At the community level, it's really education about proper hand hygiene and the like, and certainly that's paramount in hospitals too, so I shouldn't have talked about additional precautions until we focus on the hand hygiene component. But at the community level, similarly, hand hygiene, and for very resistant bugs that are readily transmitted like MRSA or community-acquired MRSA and sport teams, the issue of not sharing contaminated towels and things of that nature within the locker rooms, that's an education campaign in of itself. From the point of view of if you again have try to prevent the resistance in the first place by restricting antimicrobials. Once you actually have resistant bugs, you have to detect it with diagnosis. You have to then restrict it by using appropriate hand hygiene and precautions. Ultimately, then you're dealing with, okay, well, now we've got these resistant bugs, we're trying to contain them. How do we communicate that they're present and that people can anticipate them? That goes down to the whole surveillance and understanding what's going on ultimately. And then once you've done that, the issue is, well, how do we manage them? And the big issue there is our current pharmaceutical industries are not being given the financial incentive to continue to create in the field of infectious diseases. Drugs, for example, for cardiac disease or for other diseases that are chronic and that will require lifelong medication, they have a huge financial incentive because they know that patients are going to be on this for life. Antimicrobials are going to be given for, especially if we have our say in it, only for when they need to be done, when they need to be used, and for a short time. They're not going to be big money-making drugs for them to invest a lot of R&D into. And so without having the policies and at the regulation level, some financial incentive for these industries to create new drugs, and the pipeline being as dry as it is right now, it is a concern that ultimately we won't have the treatments needed to ultimately address these resistant organisms. And so all along from the original prevent the resistance in the first place, contain it once it happens, be able to know about it through surveillance, and ultimately be able to treat it with drugs, each and every one has a different strategy to have a solution. And it's obviously not one solution. It's multiple solutions at each one of those levels that have to be done simultaneously to really try to address this problem.